The central dogma. You will hear this phrase over and over again, and for good reason, for it entails the processes and mechanisms by which all life survives and produces copies of themselves. So, you know the classic, you go from DNA to RNA to protein. Our DNA, or an organism's DNA, stores their genetic material, and whenever a cell is needed to be copied, or another cell is needed to be duplicated, and cells are always dying, or the point is DNA needs to be copied a lot, and this process of copying is called replication. Proteins are the machinery of an organism, and they do a crazy amount. A lot of the DNA codes for protein. So the DNA is read, and using what is read, it details the structure of the protein. But an organism doesn't go directly from DNA to protein. It uses an intermediary molecule, which is RNA. So when you make RNA from a strand of DNA, this process is called transcription. Once an organism makes that RNA, it can be read as like a blueprint for the protein. And this step of going from RNA to the protein is called translation. In replication, the big player involved is DNA polymerase, which will synthesize new DNA. For transcription, the big player is RNA polymerase, which will synthesize new strands of RNA from our DNA. And in translation, we have ribosomes, which will read the RNA and help produce the correct amino acid order to make the protein, along with tRNAs, also known as transfer RNAs. DNA replication is not given on a study guide, and it won't be on the exam to the best of my knowledge, so you do not need to know it. But I think that it is integral to a lot of our understanding of DNA, and I think it'd be kind of a shame not to cover it. So if you'd like to skip ahead, feel more than welcome to, but I'm going to talk a little bit about replication. Just chill out, understand it, you don't need to memorize anything. So DNA replication, in the process of replicating our genome, we go from one copy of our genome to two copies, and they should be identical to each other. So let's say that this is a portion of DNA. We see that it is in that double helix structure, and that it is double-stranded, so they're bound to each other. Now, in order to synthesize a new nucleic acid, whether it be DNA or RNA, you need a single strand of a nucleic acid from which to read, and then pair up another strand that is complementary to it, or create a strand that's complementary to it. So in order to do that, we need to separate these bonds. Let's say like these base pairs over here, these H bonds. So we need something to break them. In DNA replication, this is a protein called helicase. So DNA helicase, let's say that it breaks these H bonds going down here, and it unravels them. Now that these single strands are exposed, something can enter, and it can begin to create a nucleic acid that is complementary to each template strand. So remember that nucleic acids have directionality, and that whenever they are paired with each other, they are anti-parallel. So if this strand over here is the 5' to 3' end, the strand that was connected to it has to be complementary. So this side is the 5', and this one is the 3'. So I mean like in the anti-parallel fashion. So now that the single strands are exposed, we can begin to synthesize our nucleic acid. So remember, we can only create a nucleic acid in the 5' to 3' direction. And it has to be anti-parallel to whatever strand that is complementary to. So if this is the 5' to 3' 5' to 3' end, we cannot synthesize it this way because it has to be anti-parallel. So we would synthesize a new nucleic acid going this way. And because this is the this is the 3' to 5' end, and we need to synthesize in the 5' to 3' end, we can do it this way as well. We start at 5' to 3', like that. Cool. So in this case, this was the 5' end, this is a 3' end, this is a 5' end, and this is a 3' end. Okay, but what's actually happening? So let's say that we zoom in on this little segment of DNA. So this one. So like this much. This would be the 3' end, and this would be the 5' end. What happens is that something needs to read the bases on our template strand, find the bases that would be complementary to it, or the nucleotides that have the complementary bases, put them in, and then make the bonds. So you put in this nucleotide that you need, and then you make a bond to another nucleotide, and you make another bond to another nucleotide. Yeah. So what does that is a polymerase. Polymerases are the ones that extend or make nucleic acids. And because it's DNA we're talking about, this is DNA polymerase. We're making more DNA. So our polymerase will bind to the template strand, a single strand, like that. It'll analyze what nitrogen base is there. There's an adenine here. And then floating around the space are free floating nucleotides. And then there are 4 types then for DNA. One of them would have an adenine, another a thymine, cytosine. This one would have a guanine. And in this case, adenine. It would look at it, what is complementary to the adenine, and that would be a thymine. So it takes this nucleotide, puts in a thymine, and then a thymine will form those H bonds. There were 2 of them. And then after that, it'll read this one. It'll go on to this one. And it'll see that, oh, we need a nucleotide that has an adenine. It's a base pair of it, like that. And as it does that, it also makes a phosphodiester bond between these. So, and then it reads the G, you would need a cytosine. That would make 3 H bonds, and then the DNA polymerase is moving down like that. And as it goes, it is also making these phosphodiester bonds. So if you notice, this was a 3' to 5' end. So we started on the 5', and we only added stuff to the 3' end. So this happens with both template strands. After our DNA helicase split it apart, a DNA polymerase bound it here, bind it here, and then it moved down the line, synthesizing the new strand of DNA, and then this one bound it here. Oh man, grammar. And then it synthesized nucleic acids going this way. Okay, all right, that was a big mouthful. So long story short, we had our original DNA with the base pairs. We broke those base pairs, and now we have 2 strands. Our DNA polymerase came in, and then it matched up nucleotides that have our matching base pairs, nitrogen bases, like that. So we went from 1 strand of DNA to 2 identical strands of DNA. And as indicated by the coloring, we see that our 2, so this would be called the parent strand. And then these would be called the daughter strands. So we kept one of the original strands of DNA in each one. So DNA replication is said to be semi-conservative because you conserve half of the original DNA. And that is DNA replication. I'm very tired, and that was very wonky. Again, you don't need to know any of it for the exam, but I hope you got something out of it.